Dr. Mara Merlino is a graduate of the interdisciplinary PhD program in social psychology at the University of Nevada, Reno, where her specialization is social psychology and law. She has conducted research under the auspices of the NIJ, NSF, and others. She is currently an associate professor of psychology and sociology at Kentucky State University. Cognitive theoretical perspectives and studies of forensic document examination, including what examiners look at, for example, eye tracking. Doctor?
And while John Paul is getting that presentation up for us, oh, he's quick. Yeah. Wow. Never mind. I thought I had a minute. First, thank you, everybody, and good morning, and thank you for the invitation to be here to speak with you today about our study. I'd like to take just a minute to acknowledge my co-investigators who are not here with us today. You see them listed there. Without this wonderful interdisciplinary team, it's very difficult to do a study such as this one. Also, I would like to thank the National Institute of Justice for the very generous grant, and particularly Jerry Laporte, who's in the audience with us today, for being a very forbearing overseer of this project. Our Kentucky State University undergraduate research assistants, many of you have met them. Most of them have graduated as of May. So we'll miss them very much. And particularly thank you to our colleagues who are in the field who helped us pilot this project to get it to the position that it's in. And thank you especially to those of you who've taken your time to participate and provide us with the data that we're going to talk about to an extent today. So let's begin with talking about a little bit about what FDEs do. You all know this a lot better than I do, being members of the field yourselves. It's fairly straightforward to talk about what FDEs do. Other people have spoken about this before, but generally speaking, FDEs evaluate signatures for their suitability for comparison. They evaluate them for the type of writing, for internal consistency, and they evaluate the range of variation. Signatures are evaluated for the presence and absence of features for comparison, and then examiners conduct side-by-side comparisons of comparable portions of the question in known writings. During this process, they determine whether there are dissimilarities, similarities, absent characters, and as I say, you all know this better than I do. So really, it's very easy for us to talk about, and we know a lot about what you do. But the question is, how do you do it? And what we are beginning to recognize in our study is that you are doing this very well with some very limited information. And so we're going to talk a little bit about how you do what you do. Beginning with some of the very, very basic micro factors that we can attend to. Now, the other speakers who have come before me in this session, whose talks are very interesting, I thank you, and I love the way that all these conversations that we're having are fitting together. I'm going to take it a little bit up a step from those more micro features, and I'm going to talk about attention, some cognitive factors that we can talk about when we're trying to determine how examiners do what they do. Attention, is defined as the sustained focus of cognitive resources on information while filtering or ignoring extraneous information, and it's often a precursor or comes before all other neurological or cognitive functions. One question we can talk about is what do we attend to? Choosing where to focus dictates where we focus visual processing resources. And you can see that demonstrated. These are actually gaze plots that we get from our eye tracking system. And some of you have seen these gaze plots before. Some of you helped make these gaze plots that we're going to see today. So we can take note of where you are sending your eyes according to what catches your attention. Interestingly, on this particular set of gaze plots, which, as I said, many of you have seen before, we sort of perhaps see some of those spots, which are fixations, falling above or outside of what we would typically think that somebody should be focusing on. And that's because our fovea can be resting in one place, and within 24 degrees of where the fovea actually is pointed, we can still perceive. And so this individual is using, this is what we know as peripheral vision. And so that's why we get those spots that are not quite on the mark. So why do we attend to these things? Well, for a couple of reasons, and it depends on the characteristic of the stimulus that we're looking at. So, for example, some features are stimulus-driven. We refer to these as exogenous features, and these are factors that really grab your attention. Something about that pulls your attention to it. Another kind of factor is a goal-directed or endogenous factor. So, as examiners, you may be looking at in a very goal-directed way at specific things, and those things are what take your attention. So, I'd like to give you a couple of signatures to look at now, and I'm going to ask you to interact with me a little bit. We've got two different kinds of signatures. One is clearly text-based, the other one is clearly stylized. I love it that you were there. What are some of the questions that come to mind about these signatures when you look at them? Anybody?
What's the name on the right?
I love it that you asked me that question. That's the perfect question. And that's exactly what I was hoping somebody would ask. When people see this signature, that is exactly what they ask. And we've heard a number of examiners say, "Boy, I would really like to know the name of the person who wrote this signatures." Why? Why is that? Okay. Well, why that is that knowing a name provides a context for the subsequent analysis. And if I were to identify this person, you can clearly see that this is M. Lene Drake on the right-hand side. This one over here could be Diane, it could be Brian, it could be, just pretty much anything. Okay? But if I tell you that signature on the right-hand side was written by a guy named Boris Vasiliev, what do you see now? You see a V in there. You can see, I wish I had a laser pointer or something, but as you go on that, on the first, I'm sorry?
I'm going to have to use the trackpad, so the
the trackpad.
240
Okay.
folks on the internet there.
Sorry, guys, on the internet?
There you go, right
okay,
here.
there we are. Okay. Thank you.
Sure.
Okay, so when we see in this structure right here, you can actually begin to see a B taking shape there. And perhaps, here's the S at the end that leads us into that. And here's the structure of the V. And it comes over like that. And you can begin to actually interpret that signature given that information. What an interesting situation this creates. You're being given a context by which to examine a signature. Now, that context is really, really important in what individuals do in terms of their analyses. The difference in the way we process these two signatures can be attributed to whether we take a top-down or a bottom-up approach to the signature itself. When we're talking about a top-down, what are those? When we talk about top-down, that's what you see there on the right-hand side of the screen. At first glance, what did you see? Probably everybody saw the bicycle. Am I right? Okay. But if you take a closer look at the bicycle, you begin to perceive that it's not really a picture of a bicycle. It looks like a bicycle, but really the component parts of it are the words for the parts of the bicycle. And only until when you start taking that apart after looking at the whole image does that start to make sense to you. So that's large chunk processing. You're taking that's an interesting thing that happened to my arrows. You're taking the larger concept and you're working downward from the large concept into the finer details. Okay. Now, over here on the right-hand side, what do you see?
Rat-faced person.
Somebody's heard the rat man. No, somebody knows the rat man illusion out there. This is a very common illusion that's used in psychological research. And I was saying that context is a really important thing when you're interpreting an image like this, a stimulus out there in the environment. The experiment where this rat man image was used was one in which it was first presented, this image was presented in the context of animals. And so you would see a picture of a cat and a dog and a fuzzy bunny or whatever, and you would see this, and you would be asked to name what you were seeing. So when it was presented with a group of animals, this image was interpreted as a rat. However, when they went back and they presented this image in a series of faces, this image is interpreted as a man. Can you guys see it? How it changes back and forth? Okay. Now, this is a really important concept when we're talking about how you go about interpreting signatures. So let's look at how bottom-up and top-down processing relate to identifying the process by which a signature is interpreted. Okay. I would like at this time, we're going to do a little exercise. I'm going to give you kind of a taste of the procedures that we have used in the study that we're currently working on. And we're still in data collection, so I'm not going to give you a lot of information. But this is a procedure that we do. It's the third procedure in our protocols. And we call this the tachistoscope extended view procedure. Okay, so I'm going to explain to you how the procedure works, and then I'm going to do a couple of tachistoscope signatures with you. What's a tachistoscope first? Okay, we call it a T-scope for short, for obvious reasons, because I can't even get it out. The T-scope is a piece of equipment that's been used in psychological research for a long time. Typically, it's used to present an array of stimuli for a very brief duration, so microseconds or even milliseconds, so quickly. It's been used in subliminal research. So the stimuli are flashed up so quickly that people really don't even know that they've seen a stimuli, okay, or a stimulus. For our part, we were very generous in terms of the time that we set for you to view the signatures. We gave you a whole second to look at them as opposed to a microsecond or a millisecond. And then we asked the examiners who were working with us on the basis of that quick 1 second view to make a call. Was it genuine or simulated? Okay. This is kind of what other examiners who helped with the pilot referred to as a high pucker factor kind of a test, because you would think there's not really very much you can do on the basis of a view of 1 second. Well, I'll get to that later. So let's do this process so that we can sort of give you a taste of what was going on in the lab at the time. Now, in the actual process, we do an extended view as well. So we would show you the signature for a second. You make a call, then we would come back and give you an extended view of the signature. You could look at it for as long as you wanted to. And then we gave you the opportunity to make a second call. So that was the way that the procedure works. And as well, we had 10 signatures that we presented right side up and 10 signatures that we presented upside down. And they were signatures of various complexity. We had text-based, stylized, and mixed signatures among those. So we did 20 different signatures using this process. But I'm only going to show you the 1 second views here. So here's what I'd like you to do. The next slide that you're going to see contains what we call a fixation cross. The cross is situated in the center of the screen. Put your gaze on the fixation cross. Don't blink. 3 seconds after the fixation cross comes up, it will stay displayed for 3 seconds, and then the slide will automatically change. Look at the signature and see what you think. Is it genuine or simulated? Ready? Wow. Okay. So our follow-up question to that was, would you say that the signature is genuine or simulated? Okay. You don't need to answer that. Just hold that thought in your mind. Okay. And our follow-up question you see there on a scale of 1, not at all confident, to 4, extremely confident. How confident would you say you are in this decision? And notice that not at all confident is a fair answer. Okay. We give you the opportunity. That was the pucker factor answer. And so we would ask you that question. So let's do another one. That was our upside down or our right side up view. So now you're going to see the upside down view, and we alternated these. Ready? Okay, 3 seconds. Okay, same question. Would you say that it's genuine or simulated, and what would be your level of confidence in that decision? Okay, the next obvious question is, what can you tell on the basis of these slides? What can you tell on the basis of seeing a signature for 1 second? Okay. Flip my notes here. Okay. Now, as you were examining those signatures, could you all make out any part of the name? Yeah, somewhat in that brief 1 second view, and so I gave you both text-based signatures, so the right side up and upside down were text-based. On signature 2, okay, could you make out any part of a name?
Yes.
You could. Wow, you guys are good. Well, and you've demonstrated that actually with these data. Okay, so now I'm going to talk about, and I'm going to tell you what the data look like for those two signatures that you just saw on the basis of the tachistoscope view and the extended view. But first I'd like to kind of go through with you how we make sense out of the eye tracking data. Now, when you sit down in front of our Tobii eye tracker, it just looks like a big computer. And it's a computer screen, and we sit you 57 centimeters away from the screen. We ask you not to move or rock back and forth or do anything to change your distance from the screen as much as possible. And then we put you through a series of these tests. And on the basis of these tests, the Tobii eye tracker actually will bounce infrared lights off people's retinas, and then it records the reflection of the infrared light. So as your retina moves or your fovea moves around the screen, that is what the eye tracker is actually recording. Okay. Now, I'm going to take you through some slides that show you what we get, the unfiltered or raw data. So this is what your eyes are actually doing when you're looking at those signatures. I'm going to show you the difference between the unfiltered and the filtered data. So we have to clean up that data a little bit in the process of doing this. I'm going to show you a different way of looking at the data, a heat map. And then I'm going to show you how we select out certain areas of interest to really focus on in terms of the examination. And I'm going to show you right now why it's important that we narrow and focus. And I'm going to be watching Brent because the statisticians in the room are going to be really impressed by this. Tell me what you think, Brent. Ready? Okay. These are actual gaze plots of unfiltered data, raw data, for each of 3 participants. Okay. Now you can actually see quite a lot on the basis of what we've got here. But the one that I found really sort of overwhelming was that one over there on the left-hand side. That is 1,200 fixations. That, if you're a and Brent just went, that's a lot of data for one little signature. That is a ton of data, okay? But you can actually see the differences in how people go about doing this visual task, okay? Down here on the end, you can see the examiner in green over there on the right-hand side. I'm sorry, I'm pointing. I keep forgetting that I should be using this thing. Okay, over here on the right-hand side, examiner in the green. You can see that the fixations are fairly evenly spread across that signature. And where'd my thing go? Uh-oh. Oh, sorry. There we go. You can see that the fixations are fairly easily spread, kind of smother that signature, don't they? Okay. Examiner 2, about half as many fixations, and they're sort of stacked up in some very specific places. Examiner number 3, almost as many as examiner number 1, but again, they're stacked up in some very, very specific places. And so these are 3 different people going about this task in very different ways. Okay, here's signature number 2. And again, that examiner who's in green up there is giving us some kind of daunting and scary stuff in terms of statistics. 7,361 fixations for that examiner as that examiner was looking at those data. And not so many for the other two, but the examiners spent a lot more time on the upside down signature than they did on the right side up signature, comparatively speaking. Okay. Now, clearly, some of these fixation points or these dots that are numbered, we can actually tell not only what you looked at, but the order in which you looked at it on the basis of these plots. So that's why they're all numbered that way. Clearly, some of that information is irrelevant because if you look up here in the, let me see if I can find my cursor again. There it is. Okay. If you look up here in the corners, you can see that the examiners were kind of hitting the fact that this is a question signature, and they were paying attention to that. Not too sure what exactly was going down right here, But there are some possible explanations for that. One examiner said, well, what if I just rest my gaze somewhere, because I'll be going along and then I'll stop and think. And it will be like that gaze burns a hole in that area. So just because there's a dot there does not mean that I keep hitting that, I'm sorry, does not mean that they're actually doing something with that particular area of space. And we maybe had an instance of that there. Okay. So we screen this stuff out by using fixation or some thresholds for our fixations. Okay, we go by a velocity threshold, which simply means that where the center of one of those dots is that's displayed on the screen, anything up to 50 pixels away is counted as part of that dot. Anything over 50 pixels away, a new dot is created. And so that's how we get all those dots. Okay. We also use a duration threshold, which is anything less than 100 microseconds, very short period of time. But anything less than that 100 microseconds is not considered a fixation. Anything over that is considered a fixation. And so using these filters, that's a little better, isn't it? You can actually begin to make some sense out of all of those different dots. Okay. So again, these are the same three examiners, by the way. It's just different colors of the dots. You can see over there in examiner 1, you can still see that person is really moving gaze around quite a bit. Number 2, not so much, really focusing on some very specific areas of that signature. And number 3, again, falls somewhere in between. Now, down there at the bottom of the screen, you actually see the fixation durations for each of these examinations. We can tell when you started looking at the signature and how quickly you get out of the signature. So examiner number 1, who really smothered the signature with all those little gaze plots, or the fixations, was looking at that signature for 30 and a little bit seconds. Okay, number 2, 29 seconds and a little bit, and number 3, 31 seconds. But still, these are very, very different visual images. But you're starting to see a pattern about where people are starting to focus on these things, especially when you begin to combine the plots. Okay, here's signature number 2. Okay, a little more activity going on this one. FDE number 1 over there with all the green dots is at 132 seconds, so gave that signature a really good look. Number 2 looked at it for a minute, and again, these are the extended views now. These are not the 1 second ones. These are the extended view gaze plots. Examiner number 3 did 44 seconds. The bigger the dot is, the longer the gaze stayed in that area. Okay, that means that there was a lot of time spent on that spot. So, filtering some more, trying to interpret these data and get a better view, a better idea of what you do. These are heat maps, okay, just kind of like weather maps. You can see where the hot spots are because they're red. On the right-hand side, the far right-hand side, you actually see the unfiltered data, and you can see the effect of the noise that we had going on with all of those unfiltered fixations. Okay. On the far left-hand side, you actually see the results of the filtered gaze plots. Now you can start to see that we're beginning to really distinguish where things are happening. And in the center, you see how we actually sort of approach this analytically. The colored blocks on the center that are overlaid over the signatures are what we refer to as areas of interest. And then based on the information that we get from the gaze plots and the heat maps and the other empirical data that are recorded on the Toby system, we can really get down and specify, I want to analyze the data for this little space right here. And this helps us to understand better and narrow and focus our analyses so we really get a better idea of what is happening with these signatures. So you can see on these signatures that there was a little bit going on, a lot of stuff over there on the far, there it is, okay. A lot of stuff going on over here in the last name. There's quite a little bit of complexity. If you can look at that on your slides. Quite a bit of stuff going on here. We have some turns. We have some different structures. Different structures here. I keep doing that. Okay. But we can actually tease those out and look at them separately. We can look at only the data for the Y. We can look at only the data for the S that's in there. We can look at little subcomponents. Okay. So, overall accuracy on these things. I've given you data. I ran some brief analyses that had to do with, and now the first thing I want to say is that this should never, ever, ever be taken as proficiency data. This is not what you do. There is never going to be a time when you're given a signature to look at for a second and be asked to make a call. That's never going to happen. So these are not proficiency data. I want to make that very, very clear. Okay, but we've given you data that demonstrate the accuracy of the examiners in the over here on. Let's see if I can not manage to. Where is it? There it is. Okay, over here on this side are the tachistoscope numbers. Okay, the green bars represent the upside down signatures. The orange bars represent the right or the. The orange bars represent the upside down. Okay. Wow. That is not really very bad. Of over 1,160 calls, or 1,600 calls, I should say, 1,647 calls across all examiners, the accuracy rate combined for the tachistoscopes and the extended views was 70%. 70%. Wow. Okay. Now, we wanted to look at that and determine whether or not that there was any, there's always the chance that, it's a 50-50 toss-up if somebody's just guessing, I really don't know about this. And so we actually looked at that. We ran a kappa statistic on those numbers. And kappa is a measure of inter-rater agreement. So we actually do have the information about whether these are genuine or simulated signatures. We do have that information. So we simply took what the examiner said and compared it to what the true answer was. And kappa is a calculation that takes away the agreement by chance. And still, even so, taking away that chance agreement, there is still moderate agreement. That's not bad. That is not bad at all for folks who got a very, very brief opportunity to look at a single signature. Okay. And down below you can actually see the data that we have by trials. Okay. The orange actually represents the incorrect calls. The green represents the correct calls. And every other signature, 1-1 represents, where is it? There it is. Okay. 1-1 represents the tachistoscope view. 1-2 represents the extended view. So that accounts for a little bit of change. When you guys got the chance to look at that signature as long as you wanted to, a few of you changed your minds and moved over, and the correct call rate went up a little bit. Okay? So signature, hello? It will catch up. Signature 2-1, of course, is the upside-down signature. The tachistoscope view. The bars under 2-2 are the extended views for the upside-down signatures, and so they alternated upside down, right side up. Okay. Here's some additional view. We broke this out according to tachistoscope, an extended view, and we ran those kappa statistics again for each of those different conditions. And as you can see, there's a little bit of a difference. When we look at only the tachistoscope views, our kappa statistic shows fair agreement, still not too bad, quite a few accurate, the accuracy of almost 68% for just viewing that signature for a second goes up quite a bit when you have the opportunity to view that signature for an extended period of time. And this also is without having an opportunity to compare to any other signatures. So you guys are pretty good at this stuff. Okay. What do the implications have? How much time do I have? Am I done? 1 minute. Okay. Fast forwarding. This stuff is important for understanding how we do what we do, and for the implications for training are, I think, fairly obvious once we understand how things are done. And what the nature of the task actually is. We can become better trainers and educators of the people who come after us. Future directions, as you see here, we can speak in terms of expertise. We can look at how the comparison process is managed, and finally, we can use these data to start investigating things like the metric properties of the scales, for example. Are there any questions in the 5 seconds we have left? Okay.
Okay, I had two very quick points. One was, I know this is still a little bit ongoing. Do you still need participants, Mara?
Oh, sure.
Okay, so people should just contact
we'll
you?
have participants until as long as we have money. When the grant money runs
okay.
out, then we're out of luck. But
the question I have on the technical side relates to the gaze plots and the difference between subject 1 and subject 2, where we had a lot of fixations, and then the other one that appeared to have less. Is there any way of figuring out if the person in the middle, the one that seemed to have fewer, was perhaps processing in the periphery more so than the one on the right? In the end result being they're going to process the same information, same data, but they're not going to fixate in the same way.
We can do that by filtering. As the original gaze plots that you saw in that very, very first slide, where I was showing that there is that peripheral stuff going on. By filtering those data, those things are going to pop out at us. So, but it's really difficult unless, which we actually do in the project. We do ask examiners in the subsequent stages of the project to interpret for us what they were actually doing with that information. We can only infer at this point, but only the examiners can give us the information about what they're doing. So we actually are doing that. Thank you for those questions.
Maura, in one of the subjects, there were a couple of gaze plots that were very far off the signature. Do you know why, the reason for that, or were they just resting their eyes?
That, again, I couldn't tell you that without asking the examiner exactly what was going on at that particular time. Could be that the examiner was resting their eyes just right there. It could be the case that an examiner may be kind of taking in the whole spatial configuration of the signatures. I've kind of noticed that as a pattern that emerges out of the data, not these particular ones, but of other gaze plots that I've looked at. So, there's so much data here, so much data.
Sort of as a follow-up to that question, when there were these extraneous markings or gazes elsewhere, did they tend to be one or two examiners, or was it just mixed? I mean, were there one or two people or certain people who tended to do that more so?
This would be anecdata at this point. I don't have any empirical reason for saying this, but so this is just my impression of what I've seen in the gaze plots. It tends to be it tends to be fairly spread out, depending. We show a lot of signatures, and there may be a point in time when the examiner just you're going along and you're thinking, and all of a sudden you stop and pause, and your eyes may not be resting on anything, but you're kind of thinking about what you're seeing. And that's one of the reasons why we wait until afterwards to ask you what you're doing with those data, because by sitting and explaining, it really sort of changes the way that you're doing things in real time. And we wanted to have you have the opportunity to do what you would do, and then to come back and talk to us about it after the fact. But I think that it just sort of tends not to be specific to individuals.
I wonder if you've looked at the data for when a misleading opinion was called, and looked back at the data for the gaze plot, is there any connection perhaps between a missed opinion and the gaze plot versus the group that got the opinion correct?
I haven't gone to that extent yet. And again, these are all preliminary data. We're still gathering data, so the numbers that we have here can only be considered preliminary. But yes, we will look at that. There's just so many things we can look at with these data that are interesting.
Really interesting experiments.
Thank you.
From the experimental design perspective,
Yes.
you said these FDEs are being shown multiple signatures.
Yes, we have a number of different protocols that we do. The first protocol, we show them single signatures, and there are 30 single signatures that they look at and make calls on. Then we do a series of question known comparisons, and the examiners are asked to do 66 of those comparisons. This is the third of the protocols where we're doing this Chekistoscope extended view protocol. We break for the day because by that time we really have worn you guys out. It's a lot of work. We break, get down your information, your gaze plots from the data, and then we show you your own gaze plots the next day, and we ask you to describe to us for a subset of those 66 signatures, what did you do and how did you do it, so we get that information from you.
So from a statistical point of view, and this is also for the statisticians in the room, you always get the question is, how do you account for independence or dependence between subsequent observations or subsequent opinions? Given the fact that these things are being shown quickly, is there a sense of whether there is correlations between subsequent opinions that the FDEs provide?
I will say that in the question known comparisons, we do counterbalance. So if there is some sort of a correlational effect, it will be distributed across the various. We do 11 trials of that part 2, and so any fatigue, any sort of variability that's unexplained like that, hopefully will be distributed across the conditions.
Okay, thank you.
Okay. Thank you, everybody.